This brief video presents some of the important features of CellSim, a proprietary computing platform for growing dynamic virtual tissues and conducting experiments in drug discovery research, cancer biology, and related fields. For the best viewing experience, we recommend you set your YouTube video resolution to HD. This first slide shows some of the important features of CellSim virtual tissues. These tissues are designed to complement wet bench research by identifying gaps in the knowledge framework, and they enable researchers to conduct virtual experiments that are difficult or impossible to do in vivo. For example, cloning and copying tissue states. They incorporate, they're based on a virtual genome that incorporates bioinformatic or patient-specific data. And the bottom line here is that overall, they reduce research time and costs by supporting rapid experiment cycles and identifying more promising targets. The core processes of CellSim computing environment are shown here. Development of virtual tissues is governed by an ontogeny engine. Starting from one or a few virtual cells and their gene-level data inputs, the virtual tissues self-construct and are able to maintain homeostasis, respond appropriately to perturbation or mutation, and in some cases, virtual tissues are capable of self-repair. Now, it's also possible to work backwards, that is, to identify a virtual genome that supports a desired type of V tissue or organoid, but we're not going to focus on that in this video today. As proof of concept, Crowley-Davis research created a library of virtual cells and tissues. 6 of those are listed here, roughly in chronological order. Many of these projects were presented at scientific meetings. Now we're going to focus in the next few slides on virtual lymphocytes that are important for studying B-cell leukemia. As shown here, virtual lymphocytes differentiate according to their local signaling environment. Some of the V lymphocytes also contain an altered signaling pathway that constitutively produces STAT5B and so can drive ELPs toward a B-cell fate even in a thymic signaling environment. Here is a cluster of these virtual progenitor cells that were grown in a thymic signaling environment. Our initial studies with virtual lymphocytes suggested that STAT5 and EBF-mediated signaling could act synergistically to drive B-cell overproduction via this STAT5B pathway. In fact, this result was later borne out in studies by collaborators using transgenic mice with haploinsufficiency in EBF1 coincident with the constitutively active STAT5B pathway. As published in a 2011 paper, the authors reported that 100% of these mice rapidly developed B-cell leukemia. Slide 5 depicts a portion of the regulatory network for virtual lymphocytes, comparing the wild type here in the left panel with the STAT5B constitutively active lymphocytes. Gene 2 here represents the bypass that works around the IL-7R signaling in the shaded rectangle here, and the bypass directly produces active STAT5 that drives gene 4. Now what I'd like to do is look at a brief video clip that shows the differentiation tracked in pseudocolor, the differentiation of virtual lymphocytes along lineage commitment pathways. As they differentiate or progress along the commitment pathway, the blue or green colors deepen and the orange pseudocolor for the early lymphocyte progenitor cells is lost. Video shows the growth, differentiation, and development of early lymphocyte progenitors with an orange pseudocolor that may differentiate into T cells or B cells according to the signals they receive. So we'll run the simulation forward until the cells divide. Some cells have lost their ELP character, and we can see if they are TN2 cells. They are not yet TN2 cells, judging by their lack of green color. Run the simulation forward. Now we see some of the cells differentiating into TN2 cells. And as we continue to monitor and let the simulation run, we'll see that there are further divisions of cells and the appearance of some pro-B cells, as shown by the blue pseudocolor. All right, now in this final slide, we have a summary of the main points covered in this video. And what I'd like to do here is just simply close with one final thought. By building the computer architecture to emulate processes involved in the development of living tissues, we've created virtual tissues that can serve to complement, expedite, and guide wet bench research. Links to other CellSim virtual tissue resources and videos are available in the description below the YouTube video screen. Thank you.